And begin. Oh, you know, I see you all had this parts of these parts of the cell. Remember that? You know that? I love this. Fun. My way. Ooh, 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 let me see if I can do this. Glad you reminded me. Why didn't you remind me? We look good here. Years ago, this was about 2000. All right. Sometimes they get horrible. Just watch out. You don't know. It just comes in all those things. Look at how they roll. This is in the blood vessel. See things attaching. That's how protein. They're going to show you how. This is what I talked about before. This is a little bit about inflammation. So it's the chemicals and changing shape. What it's going to do, it's going to attract white blood cells. Imagine this is in the interior of a blood vessel, cause changes that will allow that white blood cell to literally escape and go on its way, attracted to where the damage is. This is inside the cell. The membrane is very organized. Like we see in our illustration. It's all gel. Watch what happens. Assemble. Same thing. Same deal. I need to assemble or just little roadways. This is a motor protein. But these vesicles, it's not random. They're being carried. And they just make it seem like it's walking. It's a deep. But you see, it's the mitochondria that moves. We talk about this here. This is your RNA. Make it proteins. See the proteins going on here. Look where the protein goes. It processes and packages it. This is exocytosis. Adipose. You see a shape change inside. That's the blood. Right. Just kind of squeeze it through. White blood cell. Very heavy duty stuff. Harvard did this about 25 years ago. That was their concept of what the inside of the cell. It's basically, it dovetails a little bit into what we're talking about. Who knows how accurate it is today? I do not. I've not seen an update in years. Same when I was teaching with 25 years ago. But it gives you a good idea how complex it is on the inside. I mean, we present it with these PowerPoints and my explanations very generically. There's a ton of good stuff. Yes. So, I mean, if you want, you can enhance your understanding. Whenever time I see these And I look at these things. This was the original one, this inner life of the cell, the Harvard animation. I don't even know how long that is. But that was in BioVisions was the name of it. And I'm sure it's been chopped into different pieces like that. But I'm sure it was a very, and a lot of these folks who are lecturing, if you want to listen to them, are going to be much more talented than I am. I just don't know what level. So I try to help the magic I bring is trying to explain it to you. It's hard. And many of you, when you go into working with people, just to go off tangent, got to learn to talk at people. Not down, not up, at. Not easy to do. I worked in an area with a population that was not well educated, not the population part that I was treating. A lot of them barely got out. It's very hard to explain things that are going on in their body when the word cell is foreign. So I mean, there's a knack to communicating. My wife has a way of describing it to me. She goes, big mouth. Usually with other more descriptive terminology. So here you go. Parts of the cell. I've heard that before. Okay. Inside. Cytoplasm. Okay. Solution elements, gel elements, solid elements. That's what they call it, a solid gel. Cytosol, depending, it can be liquidy. And it's like what Jello is. When you put more proteins in an area, they hold onto the water and they get a little firmer. You can make really firm Jello. The more gelatin, which is a protein, you put in. So it's a gel-like solution made of water and protein, salt, sugars. They all contribute to it. So that's really what's in there. And the nature of those chemicals, like I said, can be very harsh, and why we have to encase things in membrane. You inherently have solids. So you have liquids, you have semi-solids like gels, and you have solids. And whether they are granules, we call inclusions, glycogen, energy. Our liver cells and our skeletal muscles have that. Pigments are a lot of the parts in our brain make those neurotransmitters, and inevitably we see pigmented. They have inherent pigment in those things that they were assembling. You'll learn about those parts of the brain. You'll learn about something called the black substance or substantia nigra. And you're going to hear about the red nucleus. Those are all areas in a portion of your brain called the midbrain where this stuff is made. Fatty droplets are in there. Crystals are in there. And the organelles, the machinery. And so we misunderstand frequently, not necessarily you or me, but to understand, we talk about membrane-bound organelle. That's the unique feature of our cells. Our cells, animal cells, eukaryotic cells, plant cells, have membrane-bound organelles. It's not just those. You saw that on the animation. Ribosomes are not inherently surrounded by membrane. When they're making proteins, which is their job, and the proteins go to certain areas, okay, at that point they become bound to membrane. It doesn't necessarily mean they're encased in it, but they're temporarily bound to the membrane, as you could see in that animation. The proteins that they're making either get incorporated in the membrane or have a little opening that takes them in to this, what we call the endomembrane system, this sort of surrounding passageway throughout the cell that's all with membrane to protect what's ever being made. Fragile stuff has to be encased in membrane because it won't make it around in the cell. So it just gives you an idea. You saw some of the cytoskeleton. Some stuff is always there, and you saw examples with those microtubules, what we call microfilaments, how they can assemble in disks. It's there, so it's dynamic. And you heard the name centrioles. The microtubules, when you learned about mitosis in high school, you learned about a spindle, right? Ringing a bell? Hello? Spindle? The chromosomes all lined up. Hey, over there, we separate all that deal. It's like in the old days when we dried clothes with a clothesline. Believe it or not, my mom was I told the other class, when my mom got an anniversary present of a clothes dryer, her world was perfect. Rather than hanging clothes outside, inside. Just saying. You were a little kid in 1956. You were lucky if you had a clothes dryer. We didn't have a dishwasher. We had a dishwasher and a dish dryer. My sister was the washer, I was the dryer. Okay, we're going. Moving on. So that's just part of what those are. So those are not membrane bound, but look at the ones they are. Mitochondria. This is the first time you're starting to see the folding that I'm going endlessly about with pleated sheets and folding the paper and all that nonsense, as you saw before. This is part of it. Membranes do that as well. And the whole idea of that is to create surface area. So what you see is effectively in mitochondria, this is all about energy transformations. When we bring sugar into the cell, in the cytoplasm, it gets started. But in order to get a lot of ATP, the only place to get that is the mitochondria. The ticket for entry into the mitochondria is oxygen. That's why when we don't have oxygen, we can't make enough ATP to surface our needs. And in 3 to 6 minutes without oxygen, you're not going to be able to survive. So that's what cellular respiration is. And you'll see that these membranes have folds. The folds are to increase surface area to a relatively limited place. You have the mitochondria is a double membrane structure. The outside membrane is semi-permeable. The inside is semi-permeable, which you'll see all the folds that they call crista for surface area. Mitochondria actually are derived, as are chlorophylls in plants, from ancient doesn't describe it, from effectively the ancient ancestors of what bacteria are. Mitochondria is the only organelle other than the nucleus that has DNA. Why? Because when you use your muscles more, they need more mitochondria. When you stop using them, you need less. So it doesn't make it anymore, and because they have a lifespan, they'll go away on their own. So if you're going on a workout program, you're going to build mitochondria to support those additionally enlarged muscles that you have. So mitochondria, depending on the activity level, whether it's in your brain or in muscle, can sort of come and go and increase. Why? Because they have their own DNA and RNA and their own ribosomes. Their DNA is a circle, not that spiral staircase, even though it's the same kind of DNA in both of these areas, but the arrangement is different. Mitochondria don't undergo mitosis or meiosis. They're not a cell. They're just an organelle, a little part inside the cell, but they do what bacteria do. They call fusion. Bacteria just keep enlarging, dividing in two, enlarging, dividing in two. It's a very simple process. Rather than mitosis is a much more complicated process that goes on. And so we call it fission. And it's part of something that you'll learn about in micro called endosymbiotic theory. That means that basically whatever the original cell or cells were gobbled up bacteria and found it to be useful, and effectively, rather than destroying it, incorporated it. You want to train it with a very, very simple approach. That's not a question for this class, but it's a question that you will certainly get early on in micro. That is there. And so you can take a look at it. Here's the DNA, circular. It has its own ribosomes. The ribosomes aren't the same as our ribosomes. They're bacterial ribosomes. We go into that in micro. They have a different density. Okay, the DNA is a circle, not a spiral staircase. Okay, and look at all the folds, and you can see it better on a real micrograph. I mean, there's a lot of intricacy in there. And what it's doing, respiration is, we want to break it down to its elementary form. Why not? I'm here. You want me to stand up? See if you are. Is this? What's respiration? We take sugar, basically glucose, C6. You don't have to write this down or anything like that. Because you'll see this a lot. O6. To get into the mitochondria, it needs oxygen. It's a reversible reaction, and what it does is it generates a lot of ATP, energy, carbon dioxide, and water. The flip side of that, so effectively that's what cellular respiration is. Brings in energy sources, principally carbohydrates. When the oxygen is present, and it has to be because we're an aerobic organism, it basically generates large amounts of ATP and waste products of carbon dioxide and water, basically breaking that apart. It's a good thing. Now, understand, we basically where that sugar come from was the reverse of this process. The reverse of this process didn't use ATP, it used sunlight, the energy from sunlight or light energy. It used water, it used carbon dioxide, and in this case, it had, I just mentioned the name before, chlorophyll. Plants makes green plants green is chlorophyll, a very similar thing to a mitochondria, and they have both. And what that basically does is it makes carbohydrates, carbs, and as a bonus, gives off water. That process is called photosynthesis, very famous. The air, not only does it provide the source for the vast majority of carbohydrates that we ingest, it also provides most of the oxygen we breathe. Most of the oxygen we breathe is probably coming from the green that you can see here. Worldwide, it comes from algae, which are found in the ocean, or oceans. Because remember, our planet is basically 5/6 covered in water. So we're, ours, let's say, is more left than them, but that's part of it. It's a magic thing. I mean, that's the plants, the greenery, all those chlorophyll-containing things are basically using our waste products and creating a food source. And by the way, have oxygen. Go home and hug a plant today. If you have a dorm room, we have a plant there. Thank you. Okay, don't. Actually, have you hugged your plant? I say, have you hugged your kid today? Have you hugged your plant today? I hugged my dog this morning. Oh, what a stupid. Don't do that. So ribosomes, not membrane-bound organs. Okay? Protein synthesis, yes. Okay? They're made up of ribosome is made up of ribosomal RNA and protein. So that combination provides the platform for what's called messenger RNA to come along. And messenger RNA, as you will see, the way it's constructed, will adhere or attach to it. And effectively, it contains signaling molecules, which is not part of that's part of cell molecular biology. And those signaling molecules will tell it either to stay in the cytoplasm, we call that a free ribosome, and the proteins are going to be used inside the cell or inside other organelles, or it's going to be one of those things you saw in the animation where it attaches to membrane-bound ribosomes. And those membrane-bound ribosomes, what they do is they attach to what's called the endoplasmic reticulum, which you're going to see as part of this network we call the endomembrane system, from the nuclear membrane all the way out to the cell's membrane, protecting things that in the cell that are either going to have to found inside membrane, like the material in a mitochondria, the material in a lysosome, the material in something called a peroxisome, the Golgi apparatus. Any of those things that are in there are going to have, if there are proteins in there, or as you will, and we'll go into this in more detail, or it will have proteins that are going to be embedded in membranes, or proteins that are going to be spit out, exocytosis. Those are the ones that are associated with bound ribosomes. They're bound, but they're not surrounded by membrane. And so you can see proteins to be incorporated into any membrane in the cell, lysosomes and their cousin you'll see called peroxisomes, or exported, released from the cell. The endoplasmic reticulum, again, the endomembrane system starts, and it's sort of out of sequence in this PowerPoint, starts at the nuclear membrane, the outer portion. Again, it's a series of membranous materials, tubes, vast tubular network. And what's in there, it has two areas, and they're basically the same. Rough endoplasmic reticulum is where those ribosomes are bound. It's not rough to the touch or anything like that. I don't think anybody's ever touched it. It's so tiny. But microscopically, it's got like little, it looks like it has little dots on it. And smooth endoplasmic reticulum is just membrane. It's really important too, because membranes are made out of fats. That's you'll see where cells process fats. That's where cells do a lot of detoxification, bad stuff that we inadvertently get ourselves into. And inevitably, I mean, we're not just the best example is people consuming alcohol. Alcohol is a toxin. Let's be clear. Whatever pleasure people derive from it, and I'm not. This is not a temperance lecture. I drink rarely, but I don't like having a drink now and again. I'm not. If somebody wants to go ahead. Thursday night of last week when we were watching the football game, my son and his buddies were up at the Naval War College in Newport, Rhode Island for a big meeting. And I said, "Where are you going to watch the game?" And he said, "We're going down for bourbon and cigars." So he's covered a lot of the drinking for the family, I'm sure. There's an exotic bourbon collection. You see those on TV now that everybody's walking? Oh my God. And pardon me while I compliment him, brag about him. When they had change of command, when he gave up the command of the destroyer, which he was in command of, and to an incoming commander, his father-in-law and I got him we split $160 bottle of bourbon. How's that for a bottle? What's going on with this kind of a bottle? Little bottle. Stag bourbon. It's okay. Like if we go out to dinner tonight, we don't do the cocktail. We're not going to do that. Okay. So here are the two of them. What makes it rough are those little bumps. And you can see the distinction as you get farther away. Here they are close. And you see cells like the pancreas that make a lot of insulin, big proteins, have a ton of that. You always come to the rough first and then the smooth. There's always both. So here you can see the artist's rendition. Here's the nuclear membrane. Those are called nuclear pores. That's how the messenger RNA and the transfer RNA and the ribosomal RNA get out. That's how building blocks get in and other building blocks go out. The DNA can't get through those openings. And that outer portion of that membrane becomes the entryway to this endomembrane system. And so you can see the smooth is here in cells like the liver, which do a lot of detoxification for alcohol and other things. Smooth endoplasmic reticulum. So the rough appears rough, it's a good word, because it's studded with ribosomes that are bound. Proteins that will be secreted or used in other membranes, etc. Proteins, enter a cistern is sort of like it was a well. It's like a little cul-de-sac, and they're modified as they go through these tubes, and then eventually, when they leave, as you will see, they're encased in a vesicle, membrane container, and are carried by those motor proteins, those guys that look like they're walking in animation, to what's called the Golgi apparatus. And the Golgi apparatus is really just packaging and shipping. It takes those materials and it shortens them. It puts tails on them and heads on them. Chemically speaking, things that make them ready to be used elsewhere. It's just like, it's the same thing. Golgi apparatus, just like you order something from Amazon online. And even though they do that a lot these days, they just send you in the original packaging. Imagine them sending you a nice wireless headset, high quality wireless set of headphones, and that's how they came to you. No packaging, no box, no bubble wrap, no nothing. That's the Golgi apparatus, packing department, the mail. That's what it does. So those things don't come to you all beat up. And that's what it is. The smooth, again, it's continuous it's just a continuation of this structure of the rough endoplasmic reticulum, but it doesn't have those proteins. They're enzymes found in the plasma membrane. Okay, you might find some of them in there. Fat metabolism, basically making all these different types of lipids and combinations of lipids. It plays a role very much in processing fats. This is what does a lot in the liver, detoxifying certain chemicals. We can't help it. They're in our environment. And it's always, and you'll hear me say lots of numbers game. The more bad stuff we have, it's like if you're working in the old days around here in Pittsburgh. The first time I came here was in 52 years ago. My dad and I flew in. And I mean, even then you could still see the remnants of the smog and all the even though they were starting to get rid of all that stuff. It was still the steel city. There was always a haze from all the factories along the Mon and along the Ohio and along the Allegheny, all big steel mills. You couldn't help but be here. I mean, it's vastly different than what it was. And so drugs, pesticides, all types of stuff you come in, that's our body. As long as it's not overly abundant, we can control it. Glycogen, a storage material, will convert to glucose. And you're going to get, when we get into nerve and muscle, you're going to hear a lot about calcium. And inside your muscle cells, you have specialized areas of endoplasmic reticulum. They're like little wells, little cul-de-sac shaped wells for calcium. Calcium plus 2, ionic calcium. Without it, skeletal and cardiac muscles, they just don't work. Smooth muscles also. Skeletal and cardiac muscles, particularly lead dependent. And here's the Golgi, more sacs, modifies, concentrates, package, whatever works for you. Okay, and they have two ends. You don't have to name the cis ends and the trans end. Okay, the cis end happens to be coming from the endoplasmic reticulum, the smooth endoplasmic. The trans end or outer face is like going to that outer membrane, like the T snares and the V snares. And all of these things, modified, tagged, sorted, packaged, directs traffic, busy. And really, and here's the actual micrograph, 90,000 times magnification, more and more folds of membrane, and you have stuff coming in, being processed, and going out. And again, the whole idea, neatly arranged and packaged in membrane for its protection. That's there. And depending on its content, this is what I mean. If it goes through there, it's going to those proteins came from the bound ribosomes. They're going to 3 places. Either, we talked about it before, those secretory vesicles, like spitting out neurotransmitters to make nerves and muscles fire. Proteins that to be used outside the cell and eventually will fuse with the plasma membrane and will be exocytosed. They'll be secreted. B, containing lipids or transmembrane proteins. They become the membrane proteins that you've heard so much about already. All those transmembrane integral proteins, peripheral proteins, blah, blah, blah. Okay, insert contents directly into that destination membrane. And sometimes it could be in the membrane in one of the vesicles, or it could be a membrane somewhere else. And lastly, these are very specific lysosomes, and so too peroxisomes, that they don't mention here. Biol. Lysosomes have enzymes for digestion and defense. The membrane is protecting the rest of the cell. Again, when you cut or hurt yourself, that lysosome can't help but explode. It releases, those are nasty enzymes. They gobble up proteins. It doesn't matter where the proteins are. They're not protein specific. If they get in to your tissues, they start gobbling up. Your tissues are made out of proteins. What happens? It hurts. It burns. It's inflamed. When you break a bone, it's coming from that stuff. You go twist your ankle, oh my God, it's killing me. And then it swells up like a balloon. It hurts more. Lysosomes. And so these contain digestive enzymes. That's why they're digesting. Again, it's not specific. Oh, I'm only an enzyme that works if he's having a hamburger or she's having a chicken sandwich. No. Proteolytic means we'll eat it all. Voila, here you are, making here, goes the apparatus, and it can go different, and they show you the different places. This can be one that's in a lysosome. This can be one being secreted. Something else can go inside of a membrane. Not a great illustration. The one some I didn't mention, peroxisomes, worth their own special. Oxygen has one job. Yeah, I told you it was the ticket to get into, sort of like your pass. If you're going to the game or going to the concert. It's your pass to get into the mitochondria. That's just a way of reflecting how it's important. It has one job. When your body is gobbling up that sugar, all those covalent, remember all the dots, all the covalent bonds, all that C's and H's with a dot here and a dot here and all that stuff. Those are all electrons. Those are those nonpolar covalent. They're chock full of energy. We take the energy in those nonpolar covalent bonds and extract it to make the ATP. That leaves us with electrons. And the whole idea of those electrons, we have to have a parking place. So oxygen gas, we take a half of one of those. Let's go. It's a little hard to visualize. We take a half of an oxygen molecule just to balance the equation. And remember, oxygen has a valence of 2, right? 6 electrons in an outermost orbital needs 2 to be happy. Here are the 2. We just ate them up here. There's 2 electrons. And voila, equals O minus 2. And then what happens is, the thing that was generating that ATP is called the proton motive force. We've got a bunch of protons, hydrogen ions. We take 2 of those, and what do we make? Water, which we can get rid of fairly easily. That's what oxygen does. It's got a fancy name, it's called the terminal electron acceptor. Its whole job in the metabolic scheme of being able to generate energy is to accept, I have a need, each molecule, I have a need for 2 electrons. Come on, bring it. Give me your electrons, your fool, your guy. Liberty statue. It's a big thing in Harvard, between New York and New Jersey. You've been there? You can do the climb. Go about 25 minutes away. The whole idea of oxygen is to capture those electrons. The electrons are thrown at it. Every so often, it picks up an unpaired electron. Electrons that don't have a parking place, we have a nasty name for them. They're called free radicals. They are the foundation of modern cancer theory and aging theory. It destabilizes. It damages. The more you have of those, the more likely it's going to create aberrant cells, cancer cells. Cells deteriorate, aging cells, cells that don't function well. So the one that's the worst is called O2 minus. It's O2 with a minus sign. It's called super oxide, not so super to me, superoxide radical. Here's other of these. The peroxisome. Its job is to detoxify these highly reactive molecules, natural byproducts of cellular metabolism, can cause havoc to the cell if not detoxified. They are inherently, if you're overly exposed to oxygen, sometimes we have to give people hyperbaric oxygen. It's a real problem. Firefighters, okay, when they have tremendous smoke inhalation, carbon monoxide, you have to use hyperbaric oxygen. You're running the risk of free radicals. We don't even do it in children. Babies, they don't have the capability to be able to do that. It's been around for a long time. It uses a couple of these. We cover them in different courses. What it does is it basically detoxifies it in steps. It uses something called an oxidase, and what that is it changes it to hydrogen peroxide. The same hydrogen peroxide you might at home be putting on a cut or a bruise. H2O2. That is also has free radicals. It's just a little bit safer. And then there's another enzyme in that peroxisome called catalase, and that changes it to water. It's the reason when you put hydrogen peroxide in intact skin, it does nothing. If you put hydrogen peroxide on damaged skin with a sore, it has, those peroxisomes have spit out this catalase. What does a peroxide do? It bubbles up. Because it's doing so rapidly, that bubbling is it's rapidly converting the peroxide to water and oxygen gas. That's the bubbling. And it does so rapidly, that's why it stings. Now you know why we've got to be careful when you put peroxide on an open wound. You'll learn, and I go over this again in micro, but you want to dilute the peroxide. Just get some water out of the old Brita filter or whatever you're using, or tap water, about 1 to 10, 1 ounce to about 10 ounces of peroxide. Let's see. It's very still anti-infective. So peroxisomes also play a role in fatty acids, but that's not as relevant to us. Here's the lysosome, the so-called safe site, because in case you've gobbled up one of these bad boys, a bacteria, a virus. It was a good virus, not a great virus. Toxins. These are not things you want to ingest. Unfortunately, we ingest them. Okay. I'm not telling you to not. I mean, we all go out to eat. You have no idea what's going on. Some places it's frightening. I mean, it is scary. And so that's, that's why I have these watchwords where I'm going out someplace for a burger. Well done. Kill it. I'll live with it. When at home, it's different. So all of those things you learn about, but this is their job. Basically, the surrounding membrane, anything that comes in, joining with a lysosome, destroying these things, those mitochondria that aren't going to be replaced, they get in there, break down and release glycogen, because that provides glucose as a sugar source. When you get to bone, we will discuss bone probably as early as next week. The way that, and it's one of the ways osteoporosis occurs, but we are constantly breaking down and putting in new bone. All of your bones have been replaced twice since you've been born right now in your age. So you do not have the initial equipment. It did not come with it. You didn't have to worry about it. It's done very, very slowly over years, but they're replaced all the time. So, and then a lot in this autodigestion or autolysis, there's another name that you'll hear about down the road very shortly called apoptosis. APOPTOSIS, programmed cell death. A lot of times when we have cells that are going down the wrong path, they're like little suicide packets and dies. I mean, a lot of times they'll take things that aren't supposed to be there as something develops and get rid of them. It's like a tadpole becoming a frog. Well, the tail's not there. A lot of the cells underwent apoptosis. They're programmed to die. That's where we derive the name. So here you go. Lysosomes are all in there, light green areas where materials are being digested. Big problem. I told you about this. Lysosomal storage diseases. When some of these enzymes have mutated, that's hereditary, genetically, and don't function. This is the Tay-Sachs disease, very common. And my first wife and I were Central European Jewish descent, called Ashkenazi Jews. Jews that are in the Holy Land are known as Sephardic Jews, because they have more. Their ancestry is Moorish, Spanish, from that Mediterranean area. If you didn't know that. So Tay-Sachs, in which the patient lacks the lysosomal enzyme to break down these glycolipids, sugars and fats in the brain. Fortunately, it's like garbage. You can't get rid of the garbage. And as they accumulate, that's how the nervous system goes bad. Seizures, mental retardation, blindness, and death. Babies normal up to about 1 year. Nightmare. The endomembrane system. Organelles, endoplasmic reticulum, Golgi, secretory vesicles, lysosomes, nuclear and plasma membranes are all part of it. They work together. Produce, degrade, store, export. All true. Harmful stuff. We have a lot of safeguards. If nothing else, and thankfully we're getting near the end of this monster. If nothing else, I hope I get the point across to you how we have a lot. When things are working normally, we have a lot of protection. We have a lot of systems in our body that recognize things that are aberrant and they undergo this apoptosis or autolysis We have a lot of safeguards to detoxify. It's a matter of degree. I mean, into that little anecdote, you would think my kid, is, and believe me, on the weekends, I wonder too sometimes. But this is somebody who spent 17 out of 24 months on a destroyer, and they can't, there's just no alcohol. So I'm not too worried about being an alcoholic. But understand, this is part of the problem. When you put too many bad things, alcohols, any other substances, I'm not telling people what to do. I mean, it's just, you got to be aware of it. And it's part of the reason when somebody's sick, it's better if they don't come into class, but they're really sick. I mean, a lot of them have a call, I don't want to come in. Okay, I can deal with that. But somebody really is ill, they should stay away, because now you're going to be coughing or sneezing, particularly, somehow releasing a lot of infectious particles that really we need to be. You can get the same thing listening to this or watching this online as you can from physically being there to a certain extent. So all of that is part of it. And there's that same typical cell we've seen a zillion times. This is going to show it to you. I don't think there's a lot to this.
The secretory protein is synthesized inside the rough endoplasmic
Here it is.
Migrates through it and exits inside the vesicle. The vesicle travels through the cytoplasm to the Golgi apparatus. The Golgi apparatus modifies the protein. The protein is dispatched from the Golgi in the transport vesicle, that then fuses with the plasma membrane, releasing the protein to the outside of the cell.
Not a whole lot there. Very good. So the cytoskeleton, this internal framework you saw in the animation. So, I mean, if you want to think of it, bone, ligaments, and muscle, maybe 3 types. By diameter. The smallest, microfilaments. The largest, by diameter, microtubules. They're hollow. Both these are, when they're assembled, they're insoluble. They're tangible structures. When you disassemble them, they dissolve. Okay? So so we're talking about the form. When they're present, they are insoluble. Intermediate filaments are the strongest. They're the ones that help make up those desmosomes and those tight junctions that we were talking about. They are always inside. Remember I did that demonstration where I spiral things? That's intermediate filaments. They're fibrous in nature and structural. Thinnest, they're made of actin. You're going to see endless examples of actin, because actin is one of the proteins that's involved in muscle contractions. They're in cells, they have an interesting web that you kind of saw a little bit on the inside of the plasma membrane. They were showing you that web or that network on the animation. There's a definite construct to it. Okay, strengthens the cell, resists compression. I'll buy that. Plays a role. This is how it changes shape, pseudopods, all of those things, endocytosis, exocytosis. These are assembled and disassembled. This is the actin. It looks like, usually an example of a double string of pearls, like a string of pearls that's twisted so it looks like 2. And that's kind of what it looks like. When you take out these little individual ones, they dissolve. When you put them together, they're fibers. Intermediate, in between, tough, insoluble ropes, twisted, braided. A tetramer, 4 fibrils twisted together. Okay, result pulling, desmosomes, internal guide wire, neurofilaments and nerve cells, keratin, skin, also in desmosome. Here it looks like, here's the tetramer, very distinctive. Microtubules, largest by diameter, hollow. They're made of something called tubulin, and they're all identical subunits, assembled and disassembled. This is from that centrosome in the center of the cell. Again, a lot of organelles, this is sort of how they're tethered or attached. They're in place, but like you saw the mitochondria in that animation, they can still move. Things are not static, they're dynamic. The fact that they're held in a place doesn't mean that they can't be moving or changing shape as you look at them. And then the substances you saw, those motor proteins changing shape. Here's what it looks like. It's kind of like spiral assembly. I don't know if that, in the animation kind of hinted at that it wasn't exactly the same. And the motor proteins change shape. Tracks to move their cargo. The centrosome is near the nucleus, means cell center. The centrioles produce those microtubules, and they come from there. And down the road, we'll take a look at centrioles as the basis of cilia and flagella. Cilia and flagella, which are in our cells, flagella only in a sperm cell in the male, cilia we all have a very, very specific arrangements of microtubules. At the centrosome, it's like a pinwheel. We call this 9 plus 0. 9 of these triplets with nothing in the middle. Here you can see it here. If you count them off, there's 9, nothing in the middle. There's something in the middle, but there's no additional microtubules. Cilia and flagella, which are all about movement, okay, and microvilli, which are about surface area, are part of what we call cellular extensions. They're in the cell. It's not like a hair sticking out. Here's the cell. A cilia or a flagella is what we call an intracellular protein appendage. It's something that protrudes, but it's encased in membrane. So all those things that look like tails really aren't tails, or look like hairs aren't really hairs. They have a very fine surround of membrane that are there. So cilia extensions on certain cells, respiratory particularly, you learned about that in lab. Pseudostratified ciliated columnar epithelia should be ringing a bell. That's the hallmark epithelia of the respiratory tract. What it does, it has goblet cells, it secretes mucus, and the cilia are like little oarsmen moving it so you can trap stuff and you can expel it. Flagella are longer by nature. Again, and sperm is an odd, but it's the only place we see it. Again, that they have a 9 plus 2. That's just really important. It's very important when we look at evolution. If we look at lower ordered ciliated, a lot of those things in pond water move around by cilia. Okay, they have a 9 plus 2 pattern. So those are certain things. They have the same DNA, the double-stranded DNA in an alpha double helix spiral staircase. Those are things that are more important evolutionarily than what Darwin did. Darwin made the case, but he didn't use a great example. Okay, over time, the idea was right, the proof was not, and we have that a lot in science. But this is more tangible proof. That 9 plus 2, and you can see it here. So it's got 9 doublets. 2, 3 when I count it, and 2 in the center. 9 plus 2. So if that was a flagella in a sperm cell, or that was a flagella in some protozoan in the pond water, a tail would have the same cross section. These are the cilia. We won't do dining arms. And microvilli are just that. The digestive tract surface area comes from these finger-like extensions. Surface area. They have these tiny actin filaments, and it's a really good illustration. That's what they look like. So I mean, all of these are areas that you can absorb liquids and foods. Here is what it looks like in an electron micrograph. And I'm sure this is the last, thankfully. The largest and last organelle in our genome is a nucleus. We already know about it. Most of our cells have one nucleus. Skeletal muscle, certain areas in the bone, we'll point them out, are multinucleated. Skeletal muscle is the best example. Red blood cells they lost their nucleus because they don't need it. It's just elegantly designed to travel, to basically transport oxygen. The nuclear envelope, you saw it's a double layer. The nucleolus or nucleolus, however you pronounce it, is a nodule of material, protein and nucleic acid type material. Okay. This is a test question for the next test. Every time. I've never written a test. I asked about the structures of a cell. What is attributed to the nucleolus? It makes ribosomal RNA. Got it? That's a standard. Everybody has that test question. You now have an answer to the test. And chromatin is where the DNA is. It doesn't become chromosomes, as you will see, until we're ready to use it to either replicate, okay, or to do transcription and translation, make proteins. Here's the nucleolus or nucleolus, right? It's not in the center. Some cells have more than one. Makes ribosomal RNA, we think, the foundation of ribosomes. The chromatome is very indistinct, the chromatin. They call it, I don't know if condensed is the right term. You'll see in a second. The natural tendency for DNA is to form this very distinctive spirals we call chromosomes. Here you can see the envelope with all those nuclear pores for things to get out and continuous with the rough endoplasmic reticulum. Outer layer continuous with rough ER as ribosomes. Inner layer mesh proteins that maintain its shape. Fine. Nuclear pores, stuff. DNA can't get out. Everything else can get in and out. Good way to look at it. You can see the pores. You can see this internal layer that maintains shape. All of those things, very, very distinct. Nucleoli, yeah, ribosomal RNA synthesis, thank you. 1 or 2 per cell, good enough. Chromatin, 30% of our threads of DNA, 60% are proteins with some RNA. You don't have to know the numbers. Actually, if you look, a better way to go back. These proteins called histones basically keep the DNA from what we call supercoiling. When you see chromosomes under a microscope, like you surely did for prophase, metaphase, anaphase, and telophase when you looked at mitosis, okay, what keeps them in that shape, okay, they naturally want to be in the shape of chromosomes. What these proteins do is effectively they stretch it out. It's like taking a spring and putting little stones in between to stretch it out. When the stones go away or somehow disappear, it returns to its normal spring-like. When it's stretched out, you can't see it as distinctly. I would use the term condensation for that, but I see errors on PowerPoints all the time. So this is what it looks like. When it gets out of that, this is what a chromosome looks like. It has a little thing in the middle of it where it attaches to the spindle. We're going to get to that soon. Okay, well, centromere, we'll get there. But really, to me, these histone proteins are the ones that allow it to remain stretched out. So effectively, it doesn't. So I hate to use the term resting state, but in this case, it's not going to be actively replicating or actively doing transcription and translation. And so they call nucleosomes. The chromosomes are the condensed. I don't know where you got the term condensed out of their PowerPoint. It was stupid. The condensed state helps protect fragile chromatin threads during cell division. Okay, and supercoils. Supercoiling is like you took a rubber band, you kept twisting it and twisting and twisting it. Did you ever do that, like with one of those planes that had the propeller? Supercoil. Oh, thank God we're done. Next time, we'll take the exam. After the exam, we're going to go into basically the cell cycle. The cell cycle is prophase, metaphase, anaphase, telophase. It's not a long presentation. It's really important. So, see everybody then. Thank you. We're good. That.